Hi, everybody.
So I'm pleased to welcome Kerry Dodd now from Buglife, who's going to talk to us about how we can use the freshwater invertebrates of our rivers as sentinels for what's going on in the rivers. So over to Kerry to talk about guardians of our rivers.
Hi, I'm Kerry from Buglife Scotland, and I'm one of the Guardians of our Rivers conservation officers at Buglife. I'll be talking about our project and about the invertebrates that we survey for the Riverfly survey. Um, but first of all, I'll just give you a quick update of who Buglife are. I mean, I think quite a few of you might have seen other talks in this series by the Buglife colleagues, but I'll just give you the spiel. So, Buglife is a UK-wide charity which is set up in the year 2000 as the Invertebrate Conservation Trust. And we're the only organisation in Europe that are concerned with the conservation of all invertebrates, from bees to beetles, worms to woodlice, jumping spiders to jellyfish. And so any animals without a backbone. There are other invertebrate-based organisations, but Buglife has taken on the big responsibility of fighting for all of them at once. Buglife's main aim is to halt invertebrate extinctions and achieve sustainable populations of invertebrates in the UK. And we do this by working hard to achieve this through promoting the importance of invertebrates and raising awareness about the challenges of their survival. Inspiring others, getting them out there and seeing things up close and showing them different species that we're working with, and also shaping policy. Now, Guardians of Our Rivers. Guardians of Our Rivers started at Buglife back in the end of 2022 and is now the hub life for the Riverfly Partnership in Scotland, delivering training and support to community groups so that they can monitor the health of their local river by surveying freshwater invertebrates. The freshwater invertebrates, they're a vital link to the in the aquatic food chain, and populations are sensitive to changes in water quality, habitat diversity, water level, and flow rate, making them the standard indicator of the health of freshwater habitats. The Riverfly monitoring provides a vital and up-to-date information on how these species are faring, but also the overall status of river environments, providing rapid feedback if the water quality declines. They act as an early warning system and is vital to our understanding of the health of our rivers, which is particularly important due to climate change. Rising temperatures are enabling some species to extend their reach northwards into Scotland, while cold-tolerant species are at risk of disappearing altogether. By monitoring freshwater invertebrates, it provides essential data to track these changes and identify any action that may be needed. The health of our rivers isn't important only to ecologists and conservationists, but also to local communities. Local communities have strong and emotional and cultural ties to their watercourses, and they provide less tangible benefits such as health improving access to nature-rich environments and opportunities for recreation. So the Riverfly survey, which I'll go into more detail after I've been through the different invertebrate groups that we monitor. You can see from this slide, which is taken from the identification guide that we give our volunteers on their training day. So we start with the caddisflies, which are separated into two groups. We have the cased caddis and the caseless caddis, and as you can see, the cased caddis make a case. We have a group of upwing flies, also known as mayflies. We have four families, the mayfly, the ephemeridae, the blue wing olive, ephemeralidae, flat-bodied heptageneidae, and the olives, betidae. We have the stoneflies, they're all in one group, and then the freshwater shrimp, which are all in another group. And these 8 groups have all been chosen due to their relative sensitivity to pollution and therefore indicative of water quality of the river. Although there is some seasonal variation, it's unlikely to affect all groups at the same time. And the 8 groups all tend to remain in the same place. So if there is a decline in numbers that's quite drastic, then that's likely to be a problem with the river rather than the invertebrates migrating. and invertebrates at the bottom of the food chain. So what affects them also affects animals that are feeding on them. So we'll take a bit of a deep dive into the different groups. So the first group is caddisfly. Here you can see the life cycle. And both the cased and caseless caddis groups are in the group of the order Tychoptera, and they're named for their hairy wings. As an adult, the adults can look similar to moths, but moths have scales on their wings and caddisflies have hairy wings. And there are 200 species of caddisflies recorded in the UK. So their life cycle, some species lay their eggs under the water, others on rocks and vegetation just above the water. And they go through 5 life stages, which are known as instars. The mature larvae then fixes, if it's a cased caddis, it will fix down its case, and if it's a caseless caddis, it will have to create a pupal case. And it will take for about 2 weeks for the larvae to emerge from the pupa case as a pupa, and then to emerge. It swims to the surface and emerges, and then from the pupal skin, the adult emerges and flies to safety. So I have an example of a few different case caddisflies. Here we've got the Melanidae, so that's the one on the left. Melanidae live in still or slow flowing water on the substrate of fine sand or fine gravel. And this distinctive shield-shaped case with an inner tube helps you identify them as a Melanidae. They can be hard to spot in the riverbed or once you've taken a sample because of the material and the shape of their case. But once they start to move and jerk like fashions, you can tend to see them. The next one is the Gueridae. This makes its case from sand grains in a tube shape and then attaches larger stones like ballast to the side of its tube. And the larvae also have finger-like projections on their pronotum. So the pronotum is the segment after the head. And when the larvae retracts into the case, these projections and the head make a sort of plug which seals the larvae into its case. And these Gueridae feed by scraping algae from surfaces. And then lastly, another example is Lignephidae. This is the largest group of Tychoptera with 58 species. It's such a large group that it doesn't have a uniquely distinguishing feature that allows you to identify the larvae from its cases. And the habitats also vary amongst this family. So for most of this family genera, most genera in this family, their predominant food source is plant material from overhanging trees and emergent vegetation, and some feed on algae. So as you can see, the case caddis make their cases from material found on the riverbed. It can be plant material, sand, stone, and small shells, and it comes in a variety of shapes and sizes. And they'll undulate their abdomen within that case to circulate the water around their gills. You can see from 2 of the families that they have their head and their 3 pairs of legs sticking out of the front of the case, and they'll drag themselves along the riverbed to move. There's some examples of artists and jewelers using caddisfly to create beautiful pieces of artwork and jewelry by putting semi-precious materials and shiny materials into a tank. And if you get the right species of case caddis, you're able to create these pieces of artwork. But as well as beautiful things, some case caddis make their cases out of plastics if they're found on the riverbed. They can be incorporated into the cases, and these make the cases weaker and more easily damaged, which means that the larvae won't be as protected from this predation. Plastic is also lighter than sand grains and small stones, which can make it easier for the caddis to be washed away during heavy rain. So our next group of caddis is the caseless caddis. Again, for the Riverfly survey, we just leave it at caseless caddis, but I've got a few here to go into a bit more depth. So most caseless caddis larvae use silk threads to strain particles out of the water which they feed upon. They'll have 3 pairs of legs, and their posterior appendages have hooks So when teaching the riverfly survey, volunteers often get these caseless caddis confused with beetle larvae because they can look quite similar, but the difference is that the caseless caddis have these hooks on the end of their appendages, their hind appendages. They swim in a thrashing motion. And again, we're going to look at 3 families, just take a bit of a deeper dive. So we've got the Polycentropoididae, the non-gilled caseless caddis. They spin the silken tubes that act like snares, and they can catch prey that are drifting in the water or moving around on the substrate. Hydrocycidae, net spinners, they construct their nets in fast-flowing water, and they feed on plant material and detritus that is caught in the net. And then lastly, we've got Rhycophidae, green sedge. They don't build cases and they actively hunt on the riverbed. They hunt blackfly larvae, Simulidae, non-biting midge larvae, Coronamidae, and also the larvae of the olive, the Betidae, the upwing fly. Speaking of upwing flies are in the order Ephemeroptera, and this refers to the lifestyles of the adults, which may only live a few days depending on how active they are. The adults have no functioning mouth parts, which means that they rely on the energy that they build up in the larval stage. So river health is really important impact on how long the adults live. There are 51 species in the UK, and they're the only insect with two separate winged stages, sorry. So the eggs are laid and the nymphs hatch and go through up to 50 molts, and they'll crawl or swim to the surface and emerge as an adult. And the first stage is called a dun. This is the immature adult where the wings are cloudy, the tails and legs are shorter. And then from this emerges the mature adult with longer tail and legs and clearer wings. So these are the 4 different families that we look for in the riverfly survey. We've got the flat-bodied Heptageneidae or stone clinger, and as the name suggests, it clings to stones in fast-flowing water. It's got a really broad head, broad flat body and legs, allows it to cling to stones and the water just flows over the top of it. Next, we have Ephemeralidae, the blue wing olive. Here, the legs are a similar size to the abdomen, and the legs and tails have dark rings around them. This family overwinters as eggs, so the larvae can be found from around May to September, and they're often found in dense weeds. We've got the Ephemeridae. This is known as the mayfly or the green drake. It has feathery gills and very powerful legs and claws as it burrows into fine sediment in slower flowing water. And then we have Betidae. This has a very streamlined shape. It's also known as a speedy darter or an agile darter. It sits still and then it will zoom really fast in very darting fashion. They have short legs in comparison to the Ephemeralidae, and the larvae of the Betidae have a rapid growth rate, which means there can be several generations in a year, with adults being present throughout the year. And we have stoneflies. They are the order Plecoptera, and they're named for the hard, shiny wings of the adults. There are 34 species in the UK, and most species are found in fast-flowing waters. Adults are poor flyers. And again, similar life cycle, but for these, they don't have a pupal phase. The nymph crawl out of the water and the adults will emerge from the nymphal skin. And again, the stoneflies don't actually get separated in the riverfly survey, so I thought we'd take a quick look at the northern February red, which is the Brachyptera putata. Northern February red is a small native endemic stonefly which has a global stronghold in the Scottish Highlands. And from the 1st of February to the 31st of March, Buglife need your help to locate, photograph and report sightings of the adults. They can be found on fence posts along the river. The adults have long antennae, similar length to the body, and weak chewing mouth parts. The wings are brown in colour, and the female have these 3 bands across their wings, as well as dark wingtips, whilst the males have short wings and can either can't fly well or are unable to fly. If you think you spot an adult of the northern February red, please could you send a photo, the date, river name, and your location. That can be a grid reference or a what 3 words to scotland@buglife.org.uk. I think that link's going into the chat. And then the last group that we look at in the riverfly survey is the freshwater shrimp. The shrimp breed throughout the year, but their peak is in the spring and early summer, and the females carry broods of eggs that may be fertilized by another male. They primarily feed on decomposing plant material and algae. So on the left, we have the native shrimp, and on the right, we have an invasive non-native shrimp. So the native shrimp is the gammarus. And you can see there it's quite an orangey colour. Sometimes it can be a bit yellow. And then on the right, we have the non-native shrimp. This fully grown one has these distinctive stripes. But smaller ones don't tend to have the distinctive stripes, but they do have these projecting cones from their urosomes. So if you follow the body round, in the gammarus, this is smooth all the way around. In the dicogammarus, this is interrupted by cones at the end of its body. So there are 2 species of dicogammarus in the UK. The killer shrimp, the Dicogammarus velocis, was first discovered in 2010 in Swaffham Water in Cambridgeshire, and the demon shrimp, the Dicogammarus haemobathes, was confirmed in the River Severn in September 2012, and they're both native to the Black Sea. So on to Guardians of Rivers. So this is the project that I work on at Buglife. Guardians of Rivers, as I've said, is the knowledge that we have about freshwater and how we can use them to indicate river health. We're passing this on to community groups so they can monitor their local river. And there's 3 main reasons why people are getting involved in Guardians of Rivers. Firstly, we have restoration. Here is a picture of a tributary of the Lothian Esk, where the Guardians group worked with landowners to plant over 200 trees to increase shading and reduce water temperature. In the middle, we've got pollution. This is the main reason people get involved with Guardians. People are becoming much more aware of the water quality of rivers, the topics in the news quite often, and monitoring the impact of any pollution is a very popular reason to get involved. And then community. People come together for the training. They meet once a month to survey and they get to know each other. In this photo, it's a group that continue to survey over the winter, having a hot drink to keep warm. So what does Guardians of Our Rivers do? Riverfly survey picks up issues that might remain undetectable by statutory monitoring alone. The statutory monitoring surveys take place in the spring and summer every 3 years. The Guardians of Our Rivers groups, they're going out monthly between April and October, so they're picking up on things that would be missed by statutory monitoring alone. So at Guardians, we're working closely with SEPA to develop a procedure for when the groups detect a pollution incident. And the presence of the Guardians groups can act as a deterrent to fly tippers, but it's also passing on knowledge to passers-by. Lots of people stop and ask what they're doing, where they see them with their trays and their spoons, and their groups can let people know of the importance of river health. The monthly scores that the groups are collating are entered into a database, and this allows the gradual trends to emerge, hopefully improvements in water quality, but also showing any problems that might be long-standing. And ultimately, we hope that Guardians of Our Rivers and the Riverfly Survey will help protect rivers, ensuring good ecological status. So how does it work? We deliver a training of the Riverfly Monitoring Initiative. That's the method that we use. It's a simple and standardized monitoring technique which is used to detect a decline in water quality. And this initiative was developed by the Riverfly Partnership in partnership with anglers, statutory bodies, conservationists and entomologists. We deliver the training in two parts. We do a theory, which is over Zoom, and then we have a practical session in person. These are both around 3 hours. In the practical session, we show the groups how to take a kick sample. You can see there my colleague with a net. When we get in the water, we have the net downstream of us. We kick in the riverbed to agitate the substrate, and then the idea is that the invertebrates let go from the substrate, and the finer bits of substrate and the invertebrates flow into the net. This is a 3-minute kick sample followed by a 1-minute hand search. Those 3 minutes are divided proportionally over the site. So rather than just standing in one place kicking for 3 minutes, we're dividing that 3 minutes over to represent the different habitats of the stretch of river that we're monitoring. And this is exactly the same method that the statutory bodies like SEPA and the Environment Agency and Natural Resources Wales are using. The volunteers, after they've done that, they put their sample into trays. You can see here the volunteers have their sample in 2 trays, and then they have this circular divided dish with a mat underneath with the 8 different groups that are our target invertebrate groups. They'll estimate an abundance for each group, and then based on the abundance, they get a score. In this survey, the higher the score, the better. So that indicates a better water quality. And this information, the score and the abundance, are both entered into an open access database called Cartographer. And this is a website that hosts lots of different citizen science projects. The data can be accessed by anybody. There's people from SEPA, Scottish Water, there are PhD students, lots of different people accessing that database and using it for their research. And we've been working closely with SEPA and using a tool called the River Invertebrate Classification Tool to provide our riverfly groups with the minimal acceptable score, which is known as a trigger score. If their score drops below this score, then there is a procedure for them to follow. It's indicative of a problem with the river. Other important aspects of the training are health and safety and biosecurity. So we go through lots of health and safety during the theory and the practical training, and we have both a generic and a dynamic risk assessment that the volunteers go through, as well as their PPE. We also promote the Check, Clean, Dry campaign to make volunteers aware of non-native species, how to identify them and how to stop the spread. This picture here is another Dicoagamerus, but also there are some of our groups that are concerned about stopping the spread of signal crayfish. So we'll give them advice on that as well. So we're in a new phase of the project, Guardians of Our Rivers Next Steps. Last year, we were successful with our application for lottery funding, and this meant that we were able to hire a new part-time conservation officer. And we're going to continue to support and train our volunteers, working closely with existing groups to help them expand and make connections with other groups in the same catchment. Here is a photo of a gathering of volunteer groups in the Don catchment in Aberdeenshire, came together to share their experiences and went on a site visit to one of the riverfly sites, which is at the site of the river restorations. That was really interesting. River restorations obviously constantly changing, so having this long-term data that the volunteers are providing will be really valuable for people who are doing research. So we can also connecting Guardians groups with their local schools, and together we're delivering workshops to schools. And we are also delivering a new for us part of the project where we're delivering extended riverfly survey. So rather than looking at 8 target invertebrate groups, the volunteers will be looking at 33 groups. It sounds like a really big jump, but quite a few of those groups are groups that the volunteers will be familiar with, like worms, snails, things like that. But from this, they can gain more information about water quality and the impact of silt and low flows on their site. So here is a map of showing all the sites that the volunteers are surveying in Scotland. We have a huge variety of volunteers. It includes individuals that have just come together because of their involvement in Guardians. We have farming clusters, anglers, friends groups, students groups, environmental groups, fisheries and rivers trusts, and catchment partnerships. We also have rangers from local authorities, estates, National Trust for Scotland, Scottish Wildlife Trust, and RSPB. But these dots on the map are more than just points of data. They represent people volunteering their time for training and surveying, and we have had some amazing feedback. So my favourite feedback that we've received so far is the one in the middle. This was from a group of anglers on the Don in Aberdeen, and one of them said, "This is the second best day of my life," while having a look at a mayfly larvae, which made me very happy. But I forgot to ask him what the first best day of his life is. So if you're interested and you're wanting to know how to take part, if you're in Scotland, the route is to email me, and we can let you know where your nearest Guardians of Our Rivers group is. If there isn't a Guardians of Our Rivers group near you, we can help you set up a new group for your community, help you pick a site, help recruit volunteers, and get everybody through the training. Yeah, join the training and then go out and take monthly surveys. Two people need to be at each survey, but this can be shared between a group. So even if you can only make a few surveys a year, that you're welcome to come along. And there's also no requirement to get in the water. If you don't feel comfortable getting in the water and taking that 3-minute kick sample, you can still come along to the surveys and help identify the invertebrates. And we provide ongoing support with data verification. We answer any of your questions, and we also host webinars. We hope to do 2 a year where we get volunteers together to talk about their experiences and share what they've learned. In the rest of the UK, the best thing to do is to email the Riverfly Partnership, and they can put you in touch with your nearest hub, which again will put you in touch with your nearest group and let you know of training opportunities. And again, going out and participating in monthly surveys and receiving that ongoing support from your hub. So I'd like to say thank you. Thank you to all of our volunteers. Firstly, we have lots and lots of amazing volunteers going out every month. And thank you to our funders. And thank you to Kieran for inviting me to talk.